Welcome back to Biomindscape. Here we can see the chip-on-chip, that is chromatin immunoprecipitation on microarray. Here what happened, it is combination of the chromatin immunoprecipitation chip with the DNA microarray analysis to identify where a protein of interest POI such as the transcription factor or modified histone binds on the genome. So first there are various steps for this. So first step is the protein bind DNA, that is the protein of interest is bound to the specific region of the genomic DNA inside the cell. Example, a transcription factor regulating gene expression. Then cross-linking and shearing, where the cell are treated with formaldehyde to cross-link fix the protein to the DNA. So the chromatin is then sheared into a small DNA fragments by sonication or enzymatic digestion. What is the purpose of it? To keep the protein DNA interaction intact while breaking DNA into a manageable pieces. Then immunoprecipitation is done and antibody specific to the POI is added. The antibody binds to the POI and pulls down the protein DNA complex, and the purpose is to isolate only DNA fragment that were bound by the protein of interest. Then next is the purifying and amplifying and labeling. The cross link to separate the DNA from the protein is done. Then purification of the DNA is done, amplification of DNA by the PCR is done, and the labeling of DNA is done by the fluorescent dye, that is green, as shown in the figure. What is the purpose of it? To prepare the DNA for detection of the microarray. Then hybridization of the DNA microarray. The fluorescently labeled DNA is added to the DNA microarray containing thousands of known DNA probes and DNA fragment hybridized with their complementary probe sequence. Then finally the detection, that is going to be happen. A scanner, it will detect the fluorescent signal on the microarray. The bright fluorescent spot indicate a genome region where the protein of interest was bound. Next step is data analysis, where the fluorescent intensity are quantified and analyzed to identify the genomic region bound by the protein of interest. Here we can see genomic locations and intensity graph, and it shows the peak that represent the enriched DNA regions bound by the protein of interest. And after that the interpretation, where we can see enhancer, promoter, gene body, and between the promoter and the gene body, that the TSS, that is transcription start site, is present, and then finally the enhancer is present. So this is the interpretation where the identified binding site of the protein of interest across the genome, like promoter, enhancer, gene bodies, etc. is shown. Here genomic wide protein DNA interaction map is given. What does chip-on-chip tell us? It identifies the binding site of the transcription factors, histone modification location, the promoter and enhancer that are regulated by the protein, and the gene regulatory networks. What is the flow chart here? Protein will bind to the DNA, cross-linking will take place, DNA sharing will take place, and then immunoprecipitation, antibody, then purification of the DNA, PCR amplification, fluorescent labeling, then hybridization to the DNA microarray, then scan the fluorescent and identify the protein binding site. So this is the chip, that is chromatin immunoprecipitation. It is used as a specific antibody against a protein of interest, and chip-on-chip is chip plus DNA microarray, that is chip-on-chip. It is used to map the protein DNA interaction across the genome, and modern studies offer use chip sequence, that is chip plus the next generation sequence, instead of chip on chip, because it provide a high resolution, greater sensitivity, and genome wide coverage. Next is the next generation sequence. So next generation sequence, what is it? Next generation sequence is a high throughput searching technique that allow millions of the DNA and RNA fragment to be sequenced simultaneously, enabling repair and accurate and cost-effective genomic analysis. First, what is done here in the NGS, that is the sample collection and DNA RNA extraction is done. High quality of genomic DNA or RNA which is converted to the cDNA. Then DNA fragmentation, that is the DNA is broken into small pieces, that is 150 to 500 base pairs, and this is done by the method of sonication or enzyme digestion or mechanical shearing. Then the end repair or A-tailing is done where the fragment ends. They are repaired and a single adenine nucleotide is added to the 3 end, that is blunt end, will be getting converted into A-tail end. At the ends at the edges we can see the A-tail. Then adapter ligation, where the adapter are ligated to both the end of each fragment. There is a P5 insert and P7 as shown in the figure. The adapter contain the P5 and P7 flow cell binding, and the primo binding site is present. Index barcode sequencing is done for multiplexing. Then library prep, where the adapter ligated fragment are purified and size selection is done to create the sequencing library. Then PCR amplification is done. The library is amplified to generate the millions of copies. The purpose is to increase the amount of DNA for the sequencing. Then clustering, the generation, that is bridge amplification is done. The fragment bind to the flow cell and they are amplified to form a cluster and identical copies. First binding, then bridge formation, then amplification cycle, and then finally we get the clusters. So million of clusters are generated and each from a single DNA fragment. Then sequencing by the synthesis like Illumina, here the fluorescent label reversible terminators nucleotide are incorporated one at a time. First in cycle 1 we can see the incorporated 1 base, then cycle 2 the image is seen, we can see here. Then cycle 3 they remove the dye and blocker. Cycle 4 everything is repeated. Now repeat will be done. A camera record the fluorescent at each cycle to determine the base. Then base calling is done. The instrument covers the fluorescent signal into the DNA sequence like ATGC. The sequence that generates a raw sequence reading on the computer. After that the bioinformatic analysis is done. The computer analyze convert the raw data into meaningful biological information like quality control. It filter the low quality readings, then read the alignments, map read out to the reference genome. Then the variation calling is happening where it identify the SNP and structural variants. Then annotation, it interpret the functional significance, and then data interpretation is done that generate the result and report. What are the application of it? It helps in the disease diagnosis and precision medicine, in cancer genomics and drug discovery, in pathogen detection and surveillance, and agriculture and plant breeding, forensic identification, metagenomics, that is microbial communities, in RNA sequencing, that is the transcriptomics, and evolution and population studies. These are the application of the NGS. Now what are the key features? Millions of reads in parallel can be known. It is fast, accurate and cost effective. It requires small amount of sample and it generates massive data for deep analysis. Common NGS platforms are Illumina, Iron Torin, PacBio and Oxford Nanopore. What are the types of NGS application? Whole genome sequencing, whole exome sequencing, targeted gene panel, RNA sequencing and chip sequencing and ATAC sequencing, etc.
DNA extraction, fragmentation, library preparation, adapters, cluster generation, sequencing, and data analysis.
Thanks for watching. Do like, share and subscribe Biomindscape.